Okay, complement. So the most important thing in the complement is that we need to know the three pathways that are important, and they kind of have different activation pathways to begin with, and they merge on a common pathway. And then again, since it's an important aspect in immune system, is a part of innate immunity. That's very interesting. It's a part of innate immunity. But without it, we cannot deal with infection. And we'll also talk of some of the anaphylatoxins that may cause anaphylaxis and some of the other functions that you will come across in terms of complement system. Okay. As I said, innate immunity is a hardwired thing, and you have to have that to fight infections. And they are components of innate immunity. And one of the lectures that we will do in the clinical part will be immunodeficiencies, where some of the players of innate immunity may be missing and you have a deficiency. So you can see from here, there are some of the important components of complement proteins. They are soluble proteins there in the blood. And C1, C2, C4, C3, and some of the controlling factors which are there. We all need them to fight infections. If we don't have one of these, for example, if you don't have C1, C2, C4, we have an immune complex disease. Again, it's hypersensitivity 2 or 3. If a person lacks C3 protein, C3 complement protein, that's a routine test that we do when we send for your complete. We look for C3. If you pay attention to the blood reports next time of yourself or the patient, you will see it says C3. So C3 is a very important complement. And if a person doesn't have that, it says susceptibility to capsulated bacteria. If a person has a deficiency from C5 to C9, the person has susceptibility to Neisseria. And Neisseria is an important bacteria. We'll discuss that in detail. And then some of the control factors which are there that control this complement protein, there are about 20 of them. And if we don't have them in the first place as a part of innate immunity, so we are susceptible to infection regardless of, there's no point giving antibiotics to these patients because they have a problem with it's just like something that doesn't come as your computer hardware and trying to upload an app would not work in this case. So 1, 2, 3, 4 important control factors and 3 important complement proteins you will see as we move along. As far as these complement proteins are concerned, they are heat sensitive. And why do we say complement? Because they complement the ability of antibodies to kill bacteria. So that's where the word complement comes from. And then again, I said earlier 20, but the list goes on and on. Sometimes we break one protein into 2 parts and give 2 different names for that. And as I said earlier, I left it for a later time after the exam 1, though it should have been in exam 1 because it's a part of innate immunity, but there's too many things to remember, cram, and I'm going to leave it on you to find out better ways. But they are important if we want to know how do we treat infection and how do we kind of gauge immunodeficiencies. Now, as I said earlier, they play a major role in defense against infectious agent. Not only that they are part of innate immunity, but even if we have a antibody-mediated acquired immune response, they are very important on those ones. And then again, since they are proteins, most of the proteins are synthesized in the liver. And if your liver function is compromised, you have infection, hepatitis, or alcoholic cirrhosis, so on and so forth. So you are unable to make those proteins, and then you will be prone to infections. But since are like, we call it like tiny bombs, they're always in the system. If you don't control them, they are going to cause problems. The question that you need to understand today is that why do we have complement proteins? And if they are there to help kill bacteria or viruses, why doesn't it kill your own cells? So our cells are very tightly protected, right? And then again, the other important thing is to keep in mind that bacteria, for example, if a bacteria divides in half an hour, one bacteria divides into two in half an hour, within a day, you will have a bacterial count as 100 trillion. You can imagine, it's just like we'll do in the lab as well. If you take 1 liter of bucket of water and you just put one bacteria over there and the water is clear, within 24 hours, it will be turbid. So you can imagine if such a thing happens in your blood. So that will be very, very serious. So we have a nature's control in terms of protecting us from infection, and that is complement, okay? But at the same time, it is tightly regulated because I said they're like tiny little bombs. They're going to blow off at any time. You don't want to kill your own cells. So what is the protective mechanisms that these complement proteins, though they are part of innate immunity? Remember we said non-specific. We also said acquired or adaptive immunity is more specific, but we still have some system in place where we may have problems in terms of controlling them if we are unable to regulate them. We divide the complement activation pathways into three: classical, lectin, and alternative pathway. And they are all different to begin with, and they merge on a common theme. So this is important that if I ask you what is the difference between these three complement pathways, the difference is in terms of how they are activated. So that's the major thing over here. And as you can see, as far as classical pathway is concerned, it is activated when one of the first complement protein that we found binds to an antigen-antibody complex. So that will bring this classical pathway in picture. The other important thing is that when you get infected with gram-positive, gram-negative bacteria or fungi or yeast, there's something that binds to the carboxy or amino terminal on the outside of those cells. And you have to have a mannan binding lectin, we call MBL, that gets activated when it binds to terminal mannose on the surface of those bacteria. So there's a good reason for the body to spot and identify those invading microorganisms before this complement protein is going to be kicked in. We call it lectin pathway. And the third is that when one of the complement protein, which is C3b, it is deposited and attached to the surface of pathogens. So this is alternative pathway, C3b, C1, and then we have MBL. So these are some of the proteins out there patrolling the body. And the moment they find infection, one of them is going to go attach to it and activate the whole series, a cascade of complement system. Okay, now let me play with it if I could. So you can see from here, as I said earlier, that there are three pathways: classical, lectin, alternative. So the important thing for you to know is that they are activated differently, And then once they are activated, they will follow the same route eventually. So then there is a common pathway that comes, but we have to understand why is it that we have three different pathways. So you can see the first one, as we said earlier, classical pathway, where you have antigen-antibody complex. There you need an antibody there. So you need an antibody for the classical pathway. And this is a bacteria. So you can see bacteria. For the lectin pathway, there is a carbohydrate attached, because most of the cell wall of microorganism is carbohydrates. But these carbohydrates are very protective. And most of our antibiotics are going to go and either break that carbohydrate outer cover. It's very difficult to break the outer shell of bacteria or mycobacteria or viruses, because your drugs cannot enter, because you have to kill the bacteria. That's why antibiotics are targeted towards that. Now, we have a system where we have mannan binding lectin that attaches to this, and then again, there will be a cascade of events that will happen. And finally, we have a C3b, which is like a hook or attachment, where it attaches to the bacteria and bring them closer to their fate in terms of being eliminated. Because as I said, most of the bacteria, it's difficult to get hold of them, kind of slippery. So the phagocytosis also, or phagocytes also struggle with that, because they tend to kind of slip out of their control. Okay. Then all of these three will converge with a common pathway, which initiates by a cleavage of C3. So that's where the rest of the pathway will begin. You have a cleavage of C3. I'm going to take each one of them in detail, but just like an overview of the serum components, for example, over here, that we can test as a blood test. And then these are complement components that are common to all pathway. Now, the most important thing that you have to remember is that all the way from complement protein 1 to complement protein 9, each one of them has a separate function, and we take them individually, and they have an important function to perform. Okay? Now, they converge on C3, and then C3 will go with different cascade of events, and I'm going to take you through how that breakdown products happen. Because one thing you have to keep as a rule is that body doesn't really keep all these either cells or any other thing in activated forms. It's like a prohormone or a naive or inactive form, because you don't want to give them a license to start blowing them up in terms of killing everything. So we need to have a control for that. That's why we have a cascade of even one after another, so we have a better control. Now what happens is that these proteins are cleaved, right, into different components. And for each component, we have different enzyme. Okay, because they are proteins. And some of them have a smaller portion, or some of them have a bigger portion. Some of the portions are there, they are soluble. Some of the portion are there for binding. Okay. Now in this case, since it's a journal graph, you can see that C3 cleaves into two parts, C3a and C3b. Of course, there's an enzyme involved. We're not going to talk at this stage, but keep in mind it's a protein. So you have to break it apart by a smaller fragment, which is A, and the bigger fragment, which is C3b. Okay, but also keep in mind that one of the complement pathways that we do for C3b is that we need C3b to attach to bacteria as an opsonin. So we heard about opsonization. So opsonization basically mean, as I said in a very late term, to sugar coat or make them attractive to phagocytes. So bacteria, you have to have a hook to attach to those outer surface like a C3b, we call like what we call a target, and then pull them up to phagocytes to phagocytose them. So that's important, C3b. But since, as I said, that they have a small doubling time, within half an hour, they're going to go more than your body cells, 100 trillion. So we want to have enormous amount of these complement proteins in our body so we can fight those infections, okay? But as we move along, you can see with every step, there is a breakdown product, right? As you go from C3, C5, and then C6 to C9, and we'll take each one of them in details, but just an overview of how the complement system works, that eventually what will happen is that the activities that these components of complement would do is that they can either be an opsonin, like C3b, and tag the bacteria. So that tag is important. And you also keep in mind that when we talked of some of the antibodies like IgM and IgG, so we said that they have a better ability to opsonize. They will go and attach to those, and they have those spaces for attachment of those opsonins. So they are basically C3b complement proteins. Okay. Then we have the smaller portions like A, we call anaphylatoxins. Again, the way the word suggests is a toxin, is going to cause anaphylaxis. But we also need a little bit of kick when we want to generate inflammation. So all those classical signs of inflammation basically are also coming from C3A and C5A. So A is a smaller portion, and these are anaphylatoxins. So C3A, C5A are anaphylatoxin, whereas C3B, just a mnemonic, B is more for binding. So B binds as compared to A, which is smaller fragment, and it floats. It goes in the circulation. And finally, we have a C6 to C9. They are there to kill pathogens. So these are some of the function, actually 3 functions, or there are many more. We just picked up 3 functions so that you have an idea what is the clinical use of this complement protein. When we go for your blood test, why do we test them? Why do we want to know, right? Because there's no point, as I said earlier, giving you antibiotics if you don't have complement proteins in the system, because we need innate immunity more than anything else. Otherwise, you will have immunodeficiencies. Okay. Now, recap of whatever I said earlier. These pathways converge onto a common pathway, which is C3, right? And then again, when it comes to C3 cleaves into C3A, I told you the anaphylatoxin, and it induces inflammatory response, whereas C3B is an opsonin. It deposits on the surface of the pathogen and make it palatable or make it attractive for the phagocyte to phagocytose them, because they are kind of slippery. Now, the 3 things that I said in the end of the figure, production of opsonin, synthesis of fluid phase molecules, which are A, C3A, C5A, and the direct killing of the pathogens. But you also have to keep in mind that these activities result either rapid removal or direct destruction of the pathogens. So they are pretty fast. They are going to go for the kill of bacteria pretty fast. And also keep in mind that if they're so fast, if they're not controlled, they are not going to kill only bacteria, they are going to kill you as well. Because the challenge is if the person's blood is teeming with bacteria, it's got bacteremia, so we have a choice. We want to kill the microorganism, but we want to save the person. But many times we employ the gadgets, and we employ the things may have a friendly fire as well. So again, self versus non-self plays an important role in terms of complement, but they are there in the system, available at all time in tons in our body to make sure that we are not infected. Because the whole point of immune system is geared towards protecting you, especially for the infection. You just receive one pinprick and that's enough for you to have hepatitis, mycobacteria, and so on and so forth. So you just have an ulcer, or you just have an abrasion. So you can see things can go out of control. The very first defense that we have in terms of innate immunity is complement. Okay, now pay attention to the first part. And as I said, that you can look at, you have ability to go back and forth. I do not, but you can look at the figure that I have for the classical pathway. And again, look at the bigger picture. You have all these activating factors that will initiate that, right? And then they follow the same route. By the end of the day, it starts from C1 and goes all the way to C9. So from C1 to C9, and the number refers to the components as they were discovered. And also just a little bit of information in terms of how we write this down in terms of our tests. The lowercase number letters present actually the breakdown cleavage products, C3A or C4B. So A is, as I said, is soluble, it floats, and the B is most of the time is binding, is a bigger chunk as compared to smaller one. Okay. Now, also, as I said earlier, keep in mind that IgG and IgM, you can go back and look at the picture of antibody. So they have those carbohydrate moieties that we talked about, the transmembrane CHO elements, and you will see over there on FC portion place for binding for C1. So you see the C1 binding places over there, okay? But in terms of activation, IgM, IgG, especially IgG3 are very effective activators of that classic pathway as compared to any other one, because type 2 or type 3, especially for lectin, MBL, or alternative, don't take the route for antibodies. So if you have deficiencies for these two antibodies, you have a problem with the classical pathway. Okay, now let's take them a little bit of detail. Okay, so let's pay attention to, for example, the classical pathway in the beginning. And remember, I was trying to show you that video so you can kind of visualize what's happening, because they're all protein, there are structures there. So a bacteria, you have initiation, so it attaches to IgG, we already talked about that. And then this is how a C1 looks like. So these are proteins, and they are peptides, and they have amino acids. So they have that structural thing. You have to keep that in mind. Different components. I don't expect you to know much detail, but just know C1. But it basically has a bouquet, like a flower bouquet kind of picture. We have different components, and all of these components will attach over here. So they can see over here, they will attach. And keep in mind, as a rule, when one complement attaches to bacteria, it calls in another complement. That's how the cascade will come. So that's, again, a series of events that you have to pay attention in terms of complement cascade. So the very first event that we want to know is an activation that happened with C1. Once it gets activated by attaching to this complex, then it brings in other players. So you can see the very first thing it brings in with a complement 4. So complement 4 then come in and breaks up into two. One is, again, a smaller fragment, C4A, and one in a bigger fragment, C4B. And I gave you a mnemonic. The B attaches, is a binding. It attaches to the bacteria. So B attaches over there. Once it comes over here, then it brings in C2, because each of them has that kind of attachment thing. It's a series of events that we have. So C2 comes over here. C2 also breaks into two. There's a C2B, and there's going to be a C2A. Let's see if I can pull it up over here. Now, what is happening in this case is that you have a bacteria, and then you have C1, and it's done activated and bringing in two important players over here. They join together because they're like protein, and this forms an enzyme. So it forms one of the most important enzymes that we have in complement system. We call C3 convertase. As again, you can see where tase is an enzyme. So if you put these together, you'll come up with a enzyme which is C3 convertase. So that's like a classical pathway that we do. Because remember, as I said, that all three will come together on C3. Right? But the way they come on the C3 is different. In this one, they have to apply complement protein 4 and complement protein 2. Now, when we move for the lectin pathway, as I said earlier, that bacteria have either a carboxy amino terminal attachment over there. So we have this carbohydrate which is going to mannan binding lectin. So there's free lectins floating in the blood. So they will spot this bacteria. They will attach those specific sites on bacteria, and then they will, this is how they look like. This is like a mannose binding lectin, and this again looks like a bouquet or like, you know, a trap, so to speak. It's a trap that's going to go and attach to those bacteria. And they're all proteins. They also pretty much do the same. They will bring in C4, it cleaves into C4H, C4B, and then they bring in C2. And again, in the end, you can see that they form an C3 convertase. So they form C3 convertase, which is the most important, but the sequence of event was different as compared to the one that we earlier saw. So that was for the lectin pathway. And finally, we have alternative pathway. Again, initiation is different. So we have a C3b, which was already produced either by other pathways or is free floating in the blood, C3b. And this C3b will attach, and you can see from here, and deposit on the cell surface, and then it will invite other factors. So they are other factors. We'll discuss that as a control factors B and D. They're important. And then they begin to kind of hatch on to the factor that initiates the C3b. So each one of them has an attachment site because they are receptors. They will, C3b will attach, and then factor B and D will attach. And these are all proteins, and they are very special configuration, and we'll explain that in a while. Now, at the same time, again, you will see that the common product in this case is a little bit different than the one that you saw earlier, where you have C3b and BB, C3BB. So if you were to put them together, you will call them C3 small letter, lowercase b with a capital letter B with a small b. So this is like a C3b capital B. So this is kind of a jargon that we normally use in clinical practice in terms of, and you can see it over here as well, C3b as a very important end product of the alternative pathway. Okay, now let's recap the lectin pathway. Again, as I said, that the gram-positive, negative, and tons of other bacteria have carbohydrate moieties outside, and they either have amino terminal or carboxy terminal, and they have terminal mannose, and we have mannose binding lectin that basically attaches to them, right? But the important thing that you need to know, if you compare, if I ask you what's the difference between the classical and lectin pathway, the difference is that it is activated in the absence of antibody. We don't have an antibody over here, right? So technically, it's a part of pure innate immune defense, but I said these are arbitrary definitions that we have for innate and adaptive. In the real picture, things get a little bit of confused or kind of overlapped in the real sense. So what's happening is that those pathogens that express the mannose, they bind to those mannose binding or mannan binding lectin. And once they bind, then they initiate this series of event in terms of complement proteins. So these are some complement proteins that are going to be initiated But the most important thing is that mannan binding lectin has to bind to that portion which is there on the bacteria. Okay, now I think is a good comparison. If I don't know whether you can see it. So you can see a classical lectin pathway, right? All of them, regardless of whatever initiated them, they pretty much ended up with this configuration over here. So they had a C3 convertase and they have C5b portion. If you compare that with the alternative pathway, so you will see alternative pathway is a little bit more confusing, but it's more in terms of attachment. The whole idea is, I mean, it's very easy for me and you to say, hey, bacteria get phagocytosed by neutrophil, but things are not that simple because in the real time, we don't see that. So what we've been seeing is that people are unable to kill this bacteria, and then we figure out that there are very specific proteins, which are plasma protein, and if you're missing those plasma proteins, you would not be able to kill, right? And you can see from here, poor little bacteria, and all these things have to be attached. It's like a tagged, right? And the question you may ask is, why is it that we want to tag it with different tags over here? Because this complement proteins have an enormous capability of killing. Had it not been the case, and they start attaching to your cells, we would all be dead. But there's a protective mechanism I'm going to tell you a little bit later. What is the protective mechanism that our cells have to avoid such a binding for complement proteins so that our cells don't get killed? But sometimes they do. Okay, that happens in many diseases, and we lose those tissues, and then we have a problem. But the important thing in this case is that you will see that each one of them, this figure tells you, because in the last figure I didn't tell you the enzymes, but now you can see these are convertases. So they are very specific enzymes, which are convertases, which are going to convert a portion of a complement into an A or B. A is going to be floating, and B is going to come and bind to that bacteria. And that happens over and over again. We have C4, C3 convertase, C5 convertase, and all these things will come over here. And also remember, when we were talking about, I think I was telling you about how the neutrophils attach to endothelium and what causes the neutrophils to penetrate those endothelium and go into the tissues. One of the things I told you that there were two receptors, and they were like in this way. They cannot hold on to the prop being presented on the neutrophil. So one of the activation of protein is you want to turn around and flip around. The word that I use at that time is that they need to polymerize. So you have to activate them so that it turns around. If I was to hold, if my hands were like this, I can't hold on to anything. They have to like this for me to hold on to, latch onto anything. So that's we call dimerization if two proteins were to do and change the configuration, or trimerization if there were three proteins. Now, right now, what you see over here is they all are randomly attaching to the different receptors on the bacteria. And you may wonder what's going on and what's going to happen eventually. But as we move along, we will answer quite a few questions that we may have in terms of the whole process that we have for phagocytosis. Okay. And for alternative pathways, some other important thing you keep in mind that this one can be triggered by any foreign substance, but again, it's an absence of specific antibody. And the most important thing that we discussed earlier as well, just a recap, important, you have to deposit C3b. Now, one thing you have to keep in mind that if you go back to the first slide, I told you there's a loop we call amplification loop. C3b is there to tag the bacteria to be killed. And we need tons of C3b there to make sure that we pretty much take control of every bacteria. Right? So we do C3b levels. So if I was to run a test on you and say send for the complete blood, I'm going to look at C3, I'm going to look at C3b. Because then I know that your immune system is normal before I give you something else. Okay? And because in many times, for example, syphilis, people sometimes are prone to some of the sexually transmitted diseases because they don't have this complement. So there's another issue. Neisseria, gonorrhea. People get gonorrhea because they don't have that specific portion of a complement protein. Okay, and some of the drugs also will chew up some of the complement protein and expose you, and you may have a higher incidence of those particular infections. Now, as far as the shared pathways is concerned, you can see from here activation of C3 and C5, and C3 cleavage basically is going to lead into two small portions. I said earlier C3a, fluid phase, C3 binds, and it's a complement activation. And then from C3 till C5 is a series of events where one complement will bind to another complement, and you have a appropriate enzyme to break it apart. So, for example, if C3 binds, so usually they say C3 is like a chainsaw. That's what they say in immunology. It's like a chainsaw. So bigger proteins come, like 5 comes, they want to chop it off into C5a, C5b. It comes 6, it's going to chop it off into C6, 8, and B. Because one of them has to be, if they are together, they are safe. They're not going to talk much. It's just like, they put this, if you see the chainsaw, they put like the cover on top of it, make sure that it doesn't go and hit you. So C3 convertase is considered in the body as a chainsaw, which is going to cut those enzymes, make them available for you to fight an infection. Okay? The important thing is that A stands for the fluid phase, B stands binds. But again, keep in mind there's always exceptions to the rule, but generally it pretty much fits most of them. So from C3 to C5, we have different components. When we come to C5b till C9, right? Now, that's what I'm going to tell you, that one of the mechanisms that our body does is that it wants to be very, very specific. And it has a capability, and we basically learned from the innate immune system how to kill bacteria, for example, because they're very hard. The cell wall is very tough. Even with the best of all the antibiotics, we're unable to penetrate the cell wall. Right? So the good thing is that we have this complement. So that's why we're working on to use body's own killing mechanism to fight an infection. So all these different components, they were lying in this direction. Now they are proteins, they align themselves into a structure which is called polymerization of protein, and they will form what we call membrane attack complex. So they will form a membrane attack complex, and that will initiate, and that's going to be like, what you call dig a hole into the cell. So these will kind of polymerize, dig a hole into the cell, and break it apart. So that is called a membrane attack complex, MAC. And we saw that in case of CD8 T cells as well. We saw this in terms of perforin as well. But this one for complement actually has a very beautiful kind of visualization that we normally see in real time. So you can see from here, as I said earlier, so these are different components being attached over here. Then we bring in all those golf-like things, but they are pointing in different directions. So they are firstly attached to the bacteria, and then they join together. And you can see from here, C5, C6, C9, they join together, polymerize, and then they form a membrane attack complex. So that's where then you can take the ions out and put the water in. So it's going to be a osmotic kill. That's what these complements do. So once they poke a hole into bacteria, all the way from C6 to C9, and if you have, for example, some of us may have deficiencies of any particular complement. If you do have, then we won't have ability to use membrane attack complex. This is called MAC. Okay, so this is what you can see attachment for all these components. Once they attach and they bind each other, they send a signal to these components from C6 to C9, and they align themselves into a structural element, which is like a what do you call a pore into the structural element. So we call membrane attack complex. Okay, and then again we don't go into immunochemistry, but you can see that it basically has components all the way from C6 to C9. Okay. Now, having said that, actually, many of our professors used to give us that joke. It just came to my mind. So it said that there was a kid, and they asked me that why 6 is scared of 7. Alfred, 6 is scared of 7 because you have 6 from C6 to C9, right? Because 7, 8, 9. Is that a point? So he used to tell us of people remember as a mnemonic that C6 to C9 are there for membrane attack complex because of that reason that 7, 8, 9. Okay? You get that? Okay. So we need to regulate that. We need to regulate that. And again, the problem in this case is that you are breaking apart all these things, and they are there. So what we have is we have many different protective elements in our body. Most of us have these markers like protective sheets, DAF, MCPC, CR1. I'll discuss that in a while. So they are found on most of mammalian cells. So we basically are protective. So they will inhibit that complement-initiated killing. So our complements, though they have this capability to kill, so when they come and try to bind to our cells, these are different activating factors or proteins that will stop that happening. And it's pretty much present on mammalian cells. And you wonder, why do we need to know that? I mean, there's another thing you have to keep in mind. Why are you teaching me that? Why do I need to know that? So one of the interesting experiments that they did was that they wanted to, of course, if you go and look at transplantation, people are on a waiting list for a long time to find the donor, exact matching donor. Then they thought, okay, let's find out any animal that we can kind of take his organ and give it to human beings. Okay, so, but before they would do that, they would do like animal experiments. So one of the experiments they did was they took like a pig's heart and gave it to a baboon heart and see if it kind of survives the graft. And guess what happened? What happened was the complement proteins in the baboon's body killed the heart of the pig. So then they found out, well, the complement protein, and they also found out the reason was because those protective mammalian surface pressure were not present on that particular, donated organ. So when we want to donate an organ, that's what we do. We want to prepare it. We want to make sure that these are not expressed. If we give it to you, your complement system, before anything, is going to kill you. That is coming as a foundational knowledge for you to understand transplantation, especially for the drugs that we have, okay? The other thing is that we have other factors which are present in fluid phase. And then the most important thing that I would want you to know is in terms of regulations are C1NH, which is a esterase inhibitor, and they is a serum protein that controls the first step for classical pathway. Then we have a C4b binding protein that we have in our body that takes care of regulating the other two pathways. And we also do the serum levels for that to make sure that they are present. Some other regulatory components that we have in our body, one of them is a serum protein factor I, and the other one is a serum factor H. and they all are there, and you can see from the complexity, but I would just accept if you know that factor I and factor H are regulators of the complement activity, and what most of the time they do is that they want to make sure that those enzymes which are released are being properly released, so we have that effect on the subsequent split. And you can see from here whether it's a classical pathway or alternative pathway. So these are the players, because over here, like factor 1, so every step from one to another cleavage step, you have the role of factor 1. So you can see factor 1, factor I is there, or factor H is there in terms of the sequence of events. So each and every is a very tightly controlled biochemical process that is there. And again, I don't mind if you know the detail, but as long as you know that they are regulated and these are the major names of the player, that's enough for you. So the functions of complement that we have talked so far are generation of opsonin. We talked about production of N-alpha-toxin and cell lysis, and we discussed that. Why do we need opsonin? Because if you don't have those opsonins like C3b and C4b, bacteria are slippery. They would never be phagocytosed, right? And also keep in mind that some of the things which are important in terms of like C3bi, we look at these levels, they do not activate complement, but they also have an opsonic activity. That's what you see mostly with the antibodies. So production of opsonin is number one. And in the real time, you can see from here, these opsonins attached to it, and then the corresponding receptors are there on the neutrophils, and the neutrophils can chew them up. The same way goes in terms of other anaphylaxis. So for example, you have this A small portion, and you need to secrete them to cause the bigger picture of inflammation, so you can bring in more players, like they are like vasodilators. They can use as a vasodilators. And you can see from here, because you need those A components to open up the spaces so that bacteria can be dealt with by different neutrophils. Okay? Okay. And these are the details of anaphylotoxins. C5a, C3a, C4a, but some of them are more important, but you can see the most important that I usually ask students is C5a as compared to C3b. So these are, as I said, vascular endothelial cell. They activate that and increase the permeability, because remember I told you that if there's inflammation, so you have to open up the veins for inflammation so that these cells can go and create that edema, create that opening up of those junctions so that we can fight. So anaphylotoxins within reasonable limits are good for you because they help us to fight those infections by creating inflammation. That's where I said the inflammation is good. The second important thing is that you have to have, as I said earlier, MAC formation, which leads to cell lysis. And another important thing I just told you for transplantation, let me give you an idea for cancer as well. If you know that in your body there's a system that can make MAC, so we also are working in terms of drug design to use this MAC complement in terms of killing your cancer cells. So there's another avenue that we have in terms of innate immune response to go after your cancer cells. Other than that, the general effects are they enhance B-cell lymphocyte responses. They control the formation and clearance of immune complexes. We talked about that in type 2 and 3. Homeostatic mechanism is that you can also remove dead cell and dying cell. And they also interact with different viruses, because we just picked up the important components, but I would want you to look at this figure and you will see how the complements are going to either take care of enhancing a B-cell response. We already talked about that because of the antibody having the site for complement. And then in terms of going after bacterial viruses, keep that in mind that most of our cells which have killing capability if you attach a small portion of fragment of complement, it basically enhances that. So it's like more of a non-specific way of enhancing the effect that we normally see. Okay, and then again, if you can go to some of the figures, you will see that some of the cells, like even antigen-presenting cells, have those specific receptors. So whenever you see CR receptors on any given cells, whether it be cells or antibodies, they are the complement receptors. And you can see from here that complement receptors are important in terms of our struggle to fight infection. And we are also learning from these complement proteins to devise new drugs for taking care of transplantation, devise new drugs for taking care of the cancer cells that we have in our body and to remove the dead tissues that we have. Okay? Any questions?